I've presented a number of posters at this meeting about the work we're doing in Brisbane. So we have an idea about cancer that is, I think, quite unique. So most people will think about cancer in terms of that you get mutations and that those mutations then drive the malignancy, okay, or cause malignancy. That's true. The second area is that people know that tumours evolve to avoid the immune system, so otherwise it wouldn't exist. If the immune system solved them, it would kill them. But any tumour, when it happens, is literally evading the immune system. We know that. So those are two big things. And the genomic instability that's inherent in a tumour, because it's got mutations, allows the tumour to evolve to avoid the immune system. And then creates the 10 characteristics, the total of 10 characteristics of Hahnemann and Weinberg, ability to invade other tissues, to spread to other sites, to avoid dying, okay, and avoid apoptosis. But we have a thing. We think there are probably a couple of other things that aren't talked about a huge amount, and one of them we think is around epigenetics. So we think that as a cancer develops, it'll switch on certain genes to survive, and it will switch other ones off. So we have identified recently a protein called human single-stranded DNA binding protein. This was identified by Professor Derek Richard, who I work with, who's my partner, my scientific partner. I'm the clinical partner in the Cancer and Aging Research Programme in Brisbane. And he discovered this in 2008, okay? And this protein is really interesting. It occurs in a subspecies called Archaea, which accounts for about a third of the biomass in the world. But because it doesn't infect us or make us sick, it doesn't cause us problems, we don't really know that much about it. But these guys, these organisms can live in boiling acid, basically, in temperatures of 90 degrees centigrade, pH 2. And so the idea is that, and because their DNA repair system is the same as humans, pretty much the same, unlike bacteria, the idea was if you could identify proteins that are involved in DNA repair in that environment, they might be really, really important. And so the protein hSSB1 is virtually unchanged in 2.5 billion years of evolution. So it means it's really important. And then we began to look at, right, okay, what happens in cancer? I mean, in every cancer, it gets upregulated. So as the cancer is transformed from normal cells to malignant, the levels of this protein rise, okay? And that's probably because the DNA is unstable, so it needs more of this to recognize and then try and fix it, right? And I think it plays a role in keeping those cells alive. And I said, right, if that does that, then are there other proteins that we could find that might be linked to this, okay? And we found a whole bunch of them. Some are downregulated and some are upregulated. And what we found is that all the proteins we've identified so far play an important role in DNA repair, and these guys are talking to each other. So today we have never presented as much data on this before at any meeting as we've done at this meeting. So we have a whole series of these proteins that we presented, and they're all linked together. And interestingly, high levels of these proteins, bar one, I'll come back to that in a second, but high levels of these proteins are associated with a worse prognosis in lung cancer. The one that's a bit different is a protein called SASH1, and it's a tumour suppressor, and in cancer it gets downregulated. We think it's switched off predominantly by being methylated, so it doesn't get expressed. And interestingly, this protein SASH1 is involved in helping when it gets downregulated, you get a reduction in the ability of the DNA to repair itself. So that's quite interesting. And if you upregulate it, you can get increased repair. And interestingly, cancer cells seem to die. So we've got not only biomarkers, but all potential targets that we can play with to maybe treat cancer. So what I presented at the meeting specifically around that in quite a bit of detail was hSSB1. So this human single-stranded DNA binding protein, unchanged for 2.5 billion years of evolution pretty much, rarely ever mutated, nearly always overexpressed in cancers. We have developed a drug to target that, and we hope to go into trial in 6 months' time. And if it works, wouldn't it be great? Because it seems to work in resistant tumours. Tumours are resistant to other therapies. The drug is under development at the moment. It's going through toxicology. We anticipate we'll have the toxicology finished in the next 3 to 4 months, and then we will go in to start a clinical trial, a phase 1, typical dose escalation trial in, we think, the third quarter of this year, provided the toxicology is okay, and at the moment it's looking good.